Hello, everyone, and welcome to the American Society for Microbiology's Microbes After Hours series. Tonight, we are going to be talking about influenza as the leaves turn and fall comes upon us. That is something that's on everyone's mind. For those of you in our audience, we will have our speakers give presentations, and then we will take questions. After that, if you will come to the microphone over on the far side of the room to ask your question. For those of you who are watching on our live stream, you can submit your questions to the Twitter feed at Microbe World or use the chat function in your video player. Microbes After Hours is a collaboration among the Communications Department, the International Affairs Department, and the American Academy of Microbiology. I'm your host for this session, Barbara Hyde, Director of Communications. We're very pleased this evening to have two outstanding flu researchers. Dr. Jeffery Taubenberger is Chief of the Viral Pathogenesis and Evolution Section of the Laboratory of Infectious Diseases at the National Institute of Allergy and Infectious Diseases at NIAID. He was previously at the Armed Forces Institute of Pathology, and when he was there, you may remember his name because he was the person who led the effort to characterize the 1918 flu virus, which in that pandemic killed approximately 50 million people worldwide, which is one of the reasons that flu is still on our radar screen today. Our second speaker, Kristen McNichol, is Associate Chief of Staff for Research at the Minneapolis VA Healthcare Center and Professor of Medicine at the University of Minnesota. And her research centers on vaccination, particularly in adults. Vaccinations are not only for kids, but adults need to get them as well. And so Dr. Taubenberger will talk about the behavior of flu virus in the wild, the ecology of the flu virus. And Dr. Nichol will talk about the clinical aspects of flu virus and why it's so important to get vaccinated and how that benefits a lot of people more than just those who are vaccinated. So we'll start with Dr. Taubenberger.
Thank you very much, Barbara. I thank the ASM and Reed and the others for the invitation to be here, and I thank you for coming. It looks like a great event. So Kristen and I thought we would do a 1-2 punch and give sort of a top 10 list of things that you didn't know about influenza that you should know. And so I'll start off and do the first 5, and then Kristen will do the second 5. So I'm going to talk about the top 5 things you should know about influenza virus biology. Set the stage for the importance of influenza in humans. So number 5 is that influenza viruses actually are not just human viruses, but are animal viruses and infect a very large number of warm-blooded animal species, both mammals and birds. And it's thought that many species of wild birds, especially waterfowl like ducks, and shorebirds are the major natural reservoir of influenza viruses, like these ducks shown in the center. And unlike influenza in humans or other mammals, it's not predominantly a respiratory virus, but mainly a GI virus shed from their cloacas, which are pictured anatomically here. Influenza viruses have a very important and interesting feature that makes them really interesting but also scary, which is that they can infect multiple species of animals, but they can actually not just cause sort of one-off infections, but they can actually stably adapt from one species of animal host to another. And this is what makes influenza such an unpredictable and dangerous and important pathogen to study for both human health and animal health. So that's the first thing that you should know about influenza. The next thing to know about it is something about the virus biology itself. So number 4 is that it has an RNA genome. That is, its genes are encoded only by RNA. It doesn't have a DNA form. It's not a retrovirus, so it never goes through DNA. It's RNA to RNA. It has a pretty small genome in which you have 8 little pieces of RNA that are called gene segments that each encode for 1 or 2 genes. In total, it has about a dozen genes. So if you think of humans have maybe 40,000 genes, the influenza virus only has about a dozen, and it does everything it needs to do with that. The very sad thing is that even though it only has about a dozen genes, we still don't know what all these proteins actually do and how they all work, so we have a lot of work still cut out for us. I'm not going to go through any more of the biology other than to say that the virus is very diverse in its genetics as well as in its protein structures, but it has it encodes for proteins that stick out like lollipops on the surface of the virus that help it bind to cells it's going to infect along the respiratory tract of mammals or the GI tract, lower GI tract in birds. It has proteins that replicate its RNA, and the virus replicates in the cells that it's going to infect. There are 2 features that are important to know. One is that as an RNA virus, it doesn't have the kind of proofreading. So like when you're typing a Word document, when you misspell the word, you get the little squiggly underlined word because it knows that you've misspelled it and asks you to correct it, or it corrects it itself. That's the way that DNA is copied. It's very difficult to induce a mutation in DNA, but in RNA it's very sloppy and there's lots of mutations made. And you would think that this would be bad, but flu has adopted a lifestyle where lots of mutations are good. So it lives kind of in a swarm or quasi-species pool of viruses with lots of different mutations. The error rate is so high that basically every single virion has at least 1 and maybe up to 10 mutations from its neighbors. And this mutational pool allows the virus to rapidly adapt to new situations, whether that's finding itself in a new host or responding to immune pressure or responding to a drug. The other thing that's important to know about the virus is since its genes are not on one little string of RNA, but 8 separate ones, if 2 different viruses infect the same host at the same time, you can get mixing and matching of the gene segments, sort of like choosing food from a Chinese menu, and you can create viruses with completely new genetic features that have very different properties from the 2 parents. And this is another very important part of influenza evolution. So in summary of where we are now, that flu viruses are present in lots of different animals, birds and mammals. Wild waterfowl are thought to be the natural host, and that there's mutation by just the accumulation of point mutations as well as this genetic mixing or reassortment. These mutations are important for allowing a virus to switch hosts, but of course you have to be able to not only just infect the host, but you have to be able to replicate efficiently in that host, and most importantly, you have to be able to be spread or that transmitted from one animal of that new host to another to sustain adaptation to a new host. So ultimately, you know, my long-term interests are to figure out how flu virus goes from this orifice to that one, and it's a complicated process that we haven't quite figured out, but the virus has. So the basic paradigms are that birds like this duck are the natural host for influenza viruses. The whole ecology of how viruses move around in species of wild birds is amazingly complex, and there are lots of experts in the room here who've spent their lives doing surveillance in wild birds, and they're learning more and more, and we still don't understand how it all works, but it's an amazing process. And then paradigm number 2 is that bird viruses adapt to pigs. And this certainly has happened, that avian viruses have stably adapted to pigs. And then the third part of the paradigm is that swine viruses adapt to humans. And outside of Washington, D.C., there has been little evidence of this up until very recently, when in 2009 there was a human pandemic that we'll talk about that actually originated as a swine-adapted virus. So it is important to understand this. So you could ask yourself for number 3, how could an animal virus become adapted to humans, and how could this virus move from an animal to a human? And there are lots of possible hypotheses, and this is one of them. I've stared at this picture for a long time, and I haven't yet decided which one is happier, but they are, I think, both very happily sharing their viruses. So what we can say is that viruses from animals have adapted to humans unpredictably and sporadically over time. It's difficult to go back through the very, very old medical literature, but in some recent work that I've done with my colleagues at NIH, we think that there have been probably at least 14 pandemics in the last 500 years occurring at unpredictable levels. There's no periodicity to this, but there have been, averaging about every 40 years or so, but that there have been 4 in the last 100 years that we should talk about. So the most recent one in 2009, which was the green arrow, where's my arrow? Yeah, green arrow, H1N1. The H in the H1N1, this refers to the two proteins on the surface of the virus, hemagglutinin and neuraminidase, and there are a whole bunch of different hemagglutinins in birds and a whole bunch of neuraminases, and depending on which ones are expressed on a virus, they're given a number, 1 through 16 for hemagglutinin and 1 through 9 for neuraminidase. So this recent swine origin pandemic was H1N1. There was a pandemic in 1968 in the red arrow caused by H3N2 virus, and then just 11 years before that in 1957, the Asian influenza pandemic was H2N2, and then back in 1918, sort of the mother of all pandemics was an H1N1 virus. And H3N2 viruses and H1N1 viruses continue to co-circulate in humans today. So if you look at pandemics and you look at mortality as one measure of the impact of a pandemic, you find that they're incredibly variable. This is looking at mortality statistics for the United States, and you can see that there is an enormous range, the 1918 having almost 700,000 deaths in the United States, and the 2009 pandemic having around 13,000 or so. So just because a virus is pandemic does not make it as lethal as the 1918 flu, but new viruses have unpredictable features, and so we don't know what the next pandemic will be like. If we look just briefly at the 1918 flu and some of the numbers, they're really outstanding and horrifying when you think about it, what influenza can do. So in the 9 months that the pandemic circulated in 1918 and early 1919, the current estimates now are that about 50 million people died worldwide. So if we can compare that over the last 25 years or so, 29 million people have died of the HIV/AIDS virus, and so almost double that died of influenza in just a few months in 1918. So it's really unbelievable when you think about that mortality level. 675,000 in the US, and if you think of a city like Philadelphia that was badly hit with the flu, had something like 16,000 deaths, of which 11,000 occurred just in 1 month, in the month of October in 1918. So that at its peak was 4,500 people died in 1 week in Philadelphia. The US suffered about 100,000 troop casualties to all causes in World War I, and 40% of those were influenza deaths. So these were healthy 18 to 25-year-old men who got influenza and died mainly in September through November of 1918. Unlike the sort of cartoon paradigms I showed, what we can say about the genetics of influenza, and I don't have time to talk about it here, but just this slide is to show that the genetics of influenza are complicated because of this ability of influenza viruses to swap genes and move around. But we now know that all the influenza pandemics since 1918 are related. And in one sense, the 1918 virus is an ancestor to all the subsequent pandemics we've had in 1957, 68, and 2009. So the 1918 flu really is sort of the mother of all the pandemics that we've had. And we've recently begun to think, David Morens and I, to use this term pandemic era, that a pandemic virus that got into humans in 1918 is still here. And so all of the subsequent influenza that we've seen and all the morbidity and mortality in humans are all related to this one virus. We don't know what happened before 1918, and we're hoping to find material that can take us further back in time. That hasn't happened yet. If you look at the impact of the 1918 flu on life expectancy in the United States, the life expectancy in the 20th century went from about 45 to 70 over the course of the century, and there was a precipitous 12 or 13-year drop in 1918, all because of influenza deaths. If you look at the pathology of people who died of influenza in 1918, something that was clear even in 1918 is that the majority of people who died actually died with bacterial pneumonias. And this is really important, that influenza by itself certainly can cause severe pneumonia, but damaging the lung and setting you up for secondary bacterial pneumonia is kind of a 1-2 punch that's extremely important. And if you think, well, that was before we had antibiotics, and of course it happened in 1918, I want to say that the pathology of fatal influenza today looks very much like this. And in the 2009 pandemic, a goodly number, perhaps even the majority of fatalities to the 2009 pandemic also had secondary bacterial pneumonias, and almost all of those people were treated with broad-spectrum antibiotics. So secondary bacterial pneumonias are extremely important in the development of severe and often fatal influenza. Here are some slides of autopsy tissues from the 1918 flu, from the collection of the Armed Forces Institute of Pathology, and in this case stained with gram stain to look at bacteria. And it's loaded with different kinds of strep and staphylococcal bacteria, and these are the same kind of bugs that cause pneumonias that we see now. One thing that I wanted to say is that pandemics vary in their mortality, and one of the features is probably that different influenza viruses have inherently different virulence properties in humans or other animals that they infect. But one thing that's become clear is that viruses like the 1918 virus and probably the H5N1 bird flu virus are not just cytopathic viruses. They're not just that the virus themselves are virulent, it's that they induce an inflammatory response that itself is really virulent. And it sort of generates what we would call an immunopathology. And there's some very good mouse and ferret and macaque data and some human data about this that would support this. And what you see in infections are that you have massive infiltration of acute inflammatory cells in the lungs of animals infected with the 1918 virus, for example, and no inflammation in the lungs of a seasonal flu virus infected animal, even though the virus replicates. So it's not lack of replication. The virus replicates, but this virus does not induce an inflammatory response, and this one induces a lethal inflammatory response. And you can measure that by the rapid upregulation of genes associated with the acute inflammatory response. So the last point that I want to make before turning it over to Kristen to talk about human influenza is that this ability of the virus to mutate is important not only for its ability to move between species, but is important for its ability to escape from pre-existing immunity, which is why the vaccines have to be reformulated all the time that she will be telling you about, but also allows the virus to very rapidly escape from antiviral drugs that are used to treat and prevent influenza, and creating big problems for the medical and public health communities. This is a family tree of the gene sequence of a hemagglutinin virus from seasonal influenza from H3N2. And the amazing thing is that each of these clumps represent a group of sequences, and that these sequences are separated just by 1 year. You can actually date influenza viruses by looking at their sequences. You can say this is a 1996 influenza virus in human, not a 1993 influenza virus, human hemagglutinin. The mutation rate is that rapid. So it's really quite remarkable. And this gives you a preview, I think, of what Kristen's talking about. These are the number of times that significant mutations have occurred in influenza virus over the last 30 years that requiring vaccine changes. And so you can see the virus continues to try to escape from pre-existing immunity. Lastly, that there are 4 approved antiviral drugs for humans to treat influenza viruses in 2 classes, and some influenza viruses circulating out there have resistance to any and all of these viruses. So recent H3 viruses are now all resistant to the adamantane drugs. Recent seasonal viruses have become resistant to the neuraminidase inhibitor oseltamivir, and the 2009 pandemic is adamantane resistance, but oseltamivir resistance, neuraminidase inhibitor resistance has been rapidly described. In our patients at the NIH, during the height of the pandemic, we have seen the emergence of multi-drug resistant influenza viruses often in less than 1 week. Especially in immunocompromised patients, we've seen really rapid development of antiviral resistance. So this is a very important problem. So we've heard about the past threats of influenza like the 1918 flu, the recent threat, the 2009 pandemic. I haven't talked much about future possible threats, but you've probably all heard about H5N1 bird flu, for which there have been several hundred infections in humans with about 60% case fatality rate. And there's a great fear that that or some other unknown virus could still adapt to humans and cause a pandemic in the future, meaning that surveillance in animals and humans and development of countermeasures against them is still extremely important. So with that, I will stop and turn it over to Kristen to continue the top 10 list.
Good evening. I appreciate the opportunity to join everyone, and I thank ASM and others for the invitation. So we've heard about the specter of pandemic influenza, and Dr. Taubenberger has given us a wonderful overview of top 5 things to keep in mind, especially with regard to pandemic influenza. I'm now going to turn about 5 top things that we should think about with seasonal influenza. So number 1, influenza is common, miserable, and life-disrupting. Even seasonal influenza is common, miserable, and life-disrupting. Annual attack rates are about 5 to 10% in adults, sometimes higher. It's a little hard to get a precise estimate. And attack rates in children are even higher, perhaps 20% or even more. That's a lot of people in this country and worldwide who become ill every year with seasonal influenza. The clinical syndrome, if you develop classic influenza, I think of as equating to pure misery. Now, what is classic influenza? It's not your common cold, and it's not the GI flu. Classic influenza is characterized by the sudden onset, and I've heard sometimes people can say almost to the second, headache, fever, cough, muscle aches that sometimes can be profound, and fatigue, and then other symptoms can be present as well. But sudden onset, fever, headache, cough, muscle aches, and fatigue. That is true misery. And the impact of this illness, when you think of tens of millions of people every year becoming ill, is huge in terms of life disruption and the misery index, if you will. We think in working populations and student populations of absenteeism or impaired work or school performance, which sometimes we call presenteeism. You're there, but you're not working at full capacity. I've never wanted to imagine my neurosurgeon being guilty of presenteeism. And increased healthcare use. Now, let me just describe for you some of the characteristics of classic influenza in adult populations. We've done some work looking at college and university students and also working adult populations, and I was just reviewing some of the numbers, and the average number of days ill for working adults who have an influenza-like illness, sort of that classic syndrome, is 8 days. They miss about 1.5 days of work if they're smart, at least 1.5 days. And then they report that they did still go back to work for 4 to 5 days before their symptoms were resolved, and about 25 to 30% seek health care. That's just in a relatively healthy working adult population. In other populations, the impact can be described even more impressively. And in fact, number 2, influenza is not only miserable and life-disrupting, but influenza can kill, and that's seasonal influenza. We've seen estimates of 50 million people dying from the 1918 pandemic flu worldwide, but seasonal influenza can also kill. I find that I used to think that, we all pretty much knew bad disease, good vaccine, and we knew what the complications of influenza are. But more recent data I've seen suggests that many of us, including healthcare providers, aren't necessarily completely aware of what some of the serious complications of influenza are. And that's probably partly because they're invisible. But the complications of influenza that are serious include bacterial pneumonia. We've seen some slides from the pandemic flu and bacterial pneumonia. But seasonal influenza also, the downstream consequences can include bacterial pneumonia and exacerbations of underlying medical illnesses. I work in the Veterans Affairs healthcare system. We have lots of patients who have chronic medical conditions that can be exacerbated if they develop influenza. And these complications can result in hospitalizations and death. In fact, in this country, there are hundreds of thousands of what we call excess hospitalizations due to the complications of influenza and tens of thousands of deaths each year. So this is a pneumonia and influenza mortality curve over time published by the Centers for Disease Control and Prevention. This is a classic curve showing death rates, in this case due to pneumonia and influenza in the red line over several years, plotted by week of the year. And one of the ways that we calculate estimated deaths due to influenza is to calculate what's called an excess death. And so these black sinusoidal lines represent what we would expect to see in the absence of epidemic seasonal influenza. And then when the red line exceeds that, when the mortality rate due to pneumonia and influenza exceeds that black line, we talk about excess deaths, and that's one way to estimate or measure the burden of influenza. This is the plot for pneumonia and influenza mortality. If you plotted all-cause mortality, it would look the same. You get this sawtooth pattern, and with the seasonality in the winter months, a good portion of that increase, we think, is caused by influenza. We estimate, and there's a lot of variability from year to year, but we estimate on average about 25,000 deaths, at least cardiorespiratory deaths that are attributable to influenza. But the range for seasonal influenza, we're now talking about 3,000 to maybe 50,000. Well, 50,000 is in the range of some of the pandemics that we've seen in the past century, and certainly not 1918. So influenza can kill. I was reminded earlier this evening that my figure here may not be perfectly explanatory. So let me tell you what this is supposed to represent. It's an iceberg, and the blue line, it represents the surface of the water. And I think of what we recognize, what we diagnose or recognize as seasonal influenza as being only the tip of the iceberg with regard to the total morbidity and mortality caused by this virus seasonally. So we recognize influenza, there are the downstream consequences, undiagnosed influenza, exacerbations of chronic diseases, secondary infections, including bacterial pneumonia, other consequences, and this results not only in misery, absenteeism, physician visits, unnecessary antibiotic use if we're treating people just with influenza, and very importantly, hospitalizations and deaths. I should also note that about half of the hospitalizations, plus or minus, occur in the elderly, but about 90 to 95% of the deaths occur in the elderly. With regard to hospitalizations, we also think of other vulnerable populations, not only people of all ages with chronic underlying medical conditions, but also very young children. In fact, very young children under the age of 2 or so, they have hospitalization rates attributable to influenza and its complications similar to those that we see in the elderly. All right, so it's a nasty illness. Number 3, we can infect others even before we feel sick. I encounter a lot of healthcare providers who don't know that. Now, influenza is transmitted by the respiratory route, and classically we think of someone coughing or sneezing, and when they cough or sneeze, but even breathing, you generate larger particles called droplets and smaller particles called aerosols. The droplets, we think, can only travel a relatively short distance, although it's not clear to me how short that is, if it's 3 feet or 6 feet, or maybe a bit farther. That's the droplet. So you want to stay at least 10 feet or so away from someone who's coughing. But the aerosol, if someone generates these very tiny particles, they can get in the air and travel a longer distance. And then there's contact. And then you go open the doorknob that I just coughed and smeared all over with whatever. So all 3 mechanisms may be important, and it's a little hard to know. Relatively speaking, we think droplet may be the predominant, but all 3 can contribute to influenza. Well, we transmit to influenza, and we transmit when we are shedding virus. This schematic tries to summarize for us viral shedding in terms of titers or amount of virus shed over time. So here's day 0, here's day 1 where you actually develop symptoms, here's day 7. And here are the symptoms. Here's illness onset in the dotted line. Now what you notice here is that the virus starts replicating and a person can shed virus before you have symptoms. In adults, we think of adults perhaps starting to shed virus for about 24 hours or so, or within 24 hours before the onset of symptoms. In children, they may start incubating and shedding that virus for even a longer period of time before the onset of symptoms. Well, how many of you stay home before you have illness symptoms? Probably not many. I generally discourage the people I work with from staying home when they're still feeling well, but it's a problem For example, if you're a healthcare provider and you haven't been vaccinated and you've become infected and you start shedding virus before you have symptoms, it's hard to know when you should stay home. And then if you look at this schematic, you can shed virus if you're an adult for 5 to 6 days or so after the onset of symptoms. Now, we sometimes recommend that people stay home until their fever is resolved or their fever is resolved for at least 24 hours. But again, at least in my world, I encounter a lot of people who don't stay home for 4 or 5 days with one influenza-like illness. So we can expose people and transmit influenza. When is influenza circulating in the community? Well, it can start about now. Typically in the upper Midwest, where I come from, we start seeing influenza starting to circulate in October, November. But influenza peaks typically in January, February, or March. So the influenza season can then extend into March, April, and sometimes into May. So we can have influenza circulating in a community for a fairly long period of time. And this also has implications, and I'll talk also about vaccination in a few moments, but this is one of the reasons why we encourage people to be vaccinated early, but to continue vaccination efforts throughout really the winter season, because influenza can still be circulating into January, February, and March. Has anybody encountered the Thanksgiving phenomenon? For years, I've seen people come in and get their influenza vaccinations before Thanksgiving, and as soon as Thanksgiving comes, demand decreases. And that's too bad, because we're missing opportunities to prevent this illness. And yes, vaccination is the best prevention for everyone. And I said for everyone 6 months of age and older, but I really should have said for everyone. We recommend flu vaccine for everyone 6 months of age and older, assuming you don't have a contraindication. But vaccination can also protect those around you. And those of us who have contact with infants should also be thinking about being vaccinated to protect those babies who aren't yet ready to mount an immune response to vaccine, which is why we don't recommend vaccinating them, at least at this time. So vaccination is the best prevention. But there's a whole menu of options for preventing and controlling influenza, and they are all important. Good hand hygiene, respiratory hygiene, and cough etiquette. When I grew up, the polite thing to do was to cough into your hand, but that's no longer the way to cough, is it? We cough like this. We avoid contaminating our hands, hopefully. Then we want to go home and wash our clothes. Contact avoidance, both if you are the person who's ill, you really do have a duty to avoid exposing others to this nasty virus. Antivirals we've heard about, and of course immunization. And let me just comment just a little bit on the contact avoidance. There was a very interesting consumer survey conducted last October 2011, where 1,044 adults were surveyed, and the results were weighted to represent the US population. And people were asked if you've ever had influenza or an influenza-like illness, and then did you go out and expose people? And 64%, nearly 2/3 said, absolutely. I go out there and I expose people. I mean, I don't stop what I'm going to be doing normally. I get out there and I expose people. We've all encountered that if you're on public transportation or if you're in the school or in your public gathering. I don't hear too many coughs now, or if you're on an airplane. About 40% said absolutely, I'd get on the airplane to travel to a business meeting. 45% said that they would generally continue all of their regularly scheduled activities. And 24% said they would even go to grandma's 80th birthday. Oh, doesn't that hurt? So we've got perhaps an educational opportunity here, but this also, I think, accurately reflects what we observe in the community, that contact avoidance is a bit tough for those of us who have things we need to do and people depend on us, and for trying to stay away from people who are sick. Options for controlling influenza. Well, we've heard about this virus that drifts and shifts and is changing all the time, and that's one reason why the vaccine is updated every single year. And of course, that's a good reason for all of us to want to be vaccinated and to seek out our vaccination every year, also to boost our immune response. And for the 2012-2013 year, we see that 2 of the 3 strains are new in the vaccine for this year. But the vaccine does typically change at least one of the 3 virus strains included most years. So it's an ever-changing vaccine to try to keep under control the ever-changing virus. Well, we still have work to do, and that's one reason why we're gathered here. This graph shows influenza vaccination trends in the United States by age group. The blue line represents for people 65 and older, one of the most vulnerable groups for serious complications of influenza. The orange line, people 50 to 64, for whom vaccination has also been recommended routinely for a number of years. And for 18 to 49 people with certain high-risk medical conditions. Even though vaccination is recommended for everybody, sometimes we tease out those groups that are especially likely to have serious complications. And so you'll see here for the elderly, vaccination rates aren't going necessarily in the right direction, and they're at about 65%. The national 2020 health goal is a vaccination rate of 90%. And we've got a significant gap there for 50 to 64. We're under 50%, and for 18 to 49s, we're under 40%. So we've got an issue here. Vaccination rates, actually, if you plotted this out for a longer period of time, going back 10 years, for example, they've largely been flat. We've made some progress in some areas. For example, healthcare workers, now we have some higher vaccination rates. And in pregnant women, we've had increased uptake. But in other groups, it's flat. And it's flat regardless of race and ethnicity, although we still have disparities in vaccine delivery. And in this graph, over a 10-year period, I'm showing you vaccination rates for white non-Hispanics in the solid line, and then other racial ethnic groups. And you can see here that we still have a challenge there as well to overcome these disparities. Well, just a few closing comments on why people do and don't get vaccinated, or why they report that they do and don't get vaccinated. These are data from a survey last year conducted by the Centers for Disease Control and Prevention, looking at why healthcare providers reported that they did get flu vaccine. And you will see here their reasons, not surprising. To protect myself was number 1. To protect my friends and family, number 2. We should all, regardless of our profession, be understanding that it does protect ourselves and it will protect those around us. And then number 3, to protect patients. Now, it turns out actually that the rationale for especially targeting healthcare workers is more of a patient safety, to protect your patients, than the first 2. But those are reasons that are offered by healthcare workers. Now, those healthcare workers who didn't get vaccinated also reported reasons. And they don't think influenza vaccines work. That's too bad. They may experience side effects, oh dear, or they don't need it. So, we do have perhaps some educational opportunities there too. For example, you cannot get the flu from a flu shot. It's an inactivated vaccine. You cannot get the flu from the flu shot. We have given hundreds of millions of doses, 1 billion doses of inactivated influenza vaccine over 10 or 15 years. And it's extraordinarily safe. Local reactions are the most commonly reported side effect after receiving a flu shot. With the live attenuated or nasal spray vaccine, that's a weakened virus as opposed to a killed or dead virus, which is the flu shot. The live attenuated vaccine may be associated with some runny nose or sore throat, which are also very mild. You cannot get the flu from a flu shot. So, improving vaccine delivery, there are opportunities for improvement. Education to providers and patients. I'm not sure everybody really understands how bad the disease is or how good the vaccines are, and there are clinical trials in many groups that clearly demonstrate the benefits of these vaccines. Systems that facilitate access, low cost, and assessment and offering a vaccine that happens automatically, for example, standing orders programs. And then recommendations from providers. For people who are healthcare providers, it should be reassuring to know how potent a healthcare provider recommendation can be even for patients who may have otherwise negative attitudes toward vaccination. In this study, where we looked at high-risk patients with negative attitudes, if with the aqua bars they didn't receive a provider recommendation, vaccination rates were low, in this case for both influenza and pneumococcal vaccination, but if they did, the provider recommendation was able to overcome those negative attitudes, and vaccination rates were about 80%. In pregnant women last year, a similar phenomenon was reported, but this has been seen in many populations. If a pregnant woman received a recommendation and the vaccine was offered in that visit, the vaccination rate was 75%. If the pregnant woman received a recommendation but didn't receive the vaccine or wasn't offered the vaccine in that visit, it may be that the practice ran out of vaccine or something, the vaccination rate dropped off, and if there was no recommendation, the vaccination rate was in the basement. So, top 5 things to know. Influenza is common, miserable, and life-disrupting. We should all want to avoid it. Influenza can also kill. We can infect others even before we feel sick. Vaccination is the best prevention, but we still have work to do. And so my advice, be sure to get your flu vaccine. Thank you.
Thank you to both of our presenters. Dr. Taubenberger and Dr. Nickel will be happy now to take your questions. Do we have a question from the internet?
Yes.
Okay. Chris, you want to read it out to me?
Shannon will.
Oh, Shannon, okay.
Hi, so we actually have a question from Natalie via Twitter, and she asks, is there a potential for a universal vaccine to be the silver bullet for influenza control in the future?
I will say that many scientists are working on that, and I will defer to my esteemed colleague for further discussion.
It's a great question. It's sort of the holy grail of influenza virus study. The problem is that viruses are so diverse, especially the proteins that are expressed on the surface of the virus, and because of its mutation rate and the diversity, there's very little in common from one virus to another, so it's very difficult to do. Many people have tried different ideas, but there is a whole new avenue of research now that in the last couple of years has become very prominent, and people have some hopes that they've found some conserved structures on the stems of the hemagglutinin proteins that might help. Emphasis on other proteins like neuraminidase could help, but at this point, it's not something that's going to happen in the next year or so, but I hope that it will eventually be possible, but it's a huge challenge.
Do we have any more questions? From anyone in the room? Please.
I'm wondering about the variant influenza virus that's certainly made headlines this past summer. And I know we don't perhaps have genetic material from swine populations to go back, but do we think this is an artifact of increased surveillance by public health labs that are now equipped to look for this, or is there something different going on out there?
Well, I will jump in and say that for sure we have enhanced surveillance and heightened awareness of some of these kinds of viruses, and we're looking more intensively. So that's part of it, but also this may represent something real.
No, well, I agree. I think it's probably a combination of things. And certainly there is more surveillance, and we need more surveillance than we have now. There's more surveillance now than has ever happened before. Let me just say as one example before I answer your direct question. If you think of the 2009 pandemic, which was a swine origin virus, we now have surveillance in animals, in wild animals, in domestic animals, and humans at an unprecedented level compared to what was available decades before. Oftentimes, genomes of these viruses after surveillance are put up in near real time on GenBank, at least within a few months. So we have tens of thousands, hundreds of thousands of influenza sequences in GenBank and other databases because of the surveillance question. The 2009 pandemic emerged in a way that we still don't actually know where the reassortment event occurred that led to this pandemic, and even if it was in a pig or a human or another animal. We don't know where on earth that reassortment event occurred, how long the virus circulated before it was documented in humans at this time of unprecedented surveillance. And you go back in time and you think, how are we ever going to figure out what happened in 1918 in the past? So the increased surveillance is certainly important, but that these viruses and this interface, the human-animal interface, is enormously important. I think that doing more surveillance can only help us understand the dynamics of that, because which of these viruses are going to go somewhere and which of them are not, and we don't know the answer to those questions yet.
And I would just say, and you may also want to comment, that the enhanced surveillance is important for early identification of these viruses and early interventions, perhaps, not only in this country, but globally.
Yeah, absolutely. There's enhanced surveillance especially in the United States and some other countries, but many parts of the world, there's really very, very little surveillance. In whole continents, there's very little surveillance going on in Africa and most parts of South America and so on. So it's a huge problem.
Any more questions from the internet or in the room? Is one in the room?
Hi. I've never gotten the flu vaccine. As far as I know, never had the flu. Is it possible that people like me could be having the flu but not really noticing it, and so I could be shedding the virus and giving it to people who are going to be miserable?
Yes. We don't know what proportion of people who are infected with the virus develop symptoms with great precision. But we estimate that 40%, maybe 50% of people who are infected may not develop symptoms. But even people who are infected may be shedding virus, perhaps for a shorter period of time, perhaps at a lower level of intensity. But I think there is reasonable evidence to suggest that even someone who has never been sick in her life should still be vaccinated to help protect yourself down the line. Or even this year, maybe you won't be so lucky in terms of developing symptoms, but also to protect those around you.
Could you discuss a little bit the age differences in susceptibility? I understand this was very stark with the 1918 viruses, with unexpectedly the younger population being infected. And could you talk about that? And do we still see this phenomenon today where people are retaining their immunologic memories, perhaps from the past?
So lots of important stuff in these questions. So I'll take a start and then you can add on. So the age susceptibility in humans to influenza viruses actually differs by influenza strain. It sort of depends on kind of the pre-existing population immunity level. So in general, young children, newborns, and young children have not been exposed to viruses at all, so they're a very susceptible population. And the elderly, who obviously have been exposed to a lifetime of influenza, often are very susceptible, not because they haven't been exposed, but because sometimes their immune system is not quite as good at responding to infections as it was previously. So in the sort of two extremes of life, you have these high-risk populations, and it sort of varies in between. But generally, influenza in humans, the sort of peak infections are in the younger half of the population. But this certainly can vary. The 2009 pandemic is an example in which you had a skewing of more susceptible people who were younger because older people had some cross-protective immunity because of the ancestors of the 1918 virus that had circulated. But that certainly can vary from outbreak to outbreak or epidemic to epidemic.
And of course, while that's very true, I think it's also important when we think about seasonal influenza to remember that everyone in every age group is potentially susceptible. We may not see the same attack rates or levels of illness from year to year in different populations, but even in working adult populations or elderly populations, if there has been very aggressive activity for trying to obtain laboratory confirmation when people develop an influenza-like illness, attack rates of 5 to 10% are described even in elderly populations.
Please, Dr. Campos.
Would each of you mind commenting on the biological and clinical differences between influenza A and B?
Well, okay, so on the biology, they are both influenza orthomyxoviruses, that they share a common evolutionary ancestry, but influenza B viruses, unlike the influenza A viruses that I talked about in my summary and that Kristen talked about in humans, are influenza Bs are basically human-specific viruses that they have occasionally been pulled out of some animals, including seals, but they're really human-adapted viruses. And they probably split off from the common influenza ancestor several thousand years ago and have become human-specific viruses. They circulate every year, cause outbreaks. They're part of the mix of annual seasonal influenza. So the fact that we haven't talked about influenza B, it's an excellent point, is not to dismiss its importance. It's part of seasonal influenza. It drifts. It has features similar to influenza A. It doesn't have pandemic potential in the sense that there's no animal reservoir to it, but it is certainly an important viral pathogen for humans.
And from the clinical perspective, we tend to think of influenza B as being particularly an influenza illness that affects children who may be particularly susceptible to influenza B, as opposed to influenza A, especially H3N2, that is particularly serious for the elderly. But again, all age groups can be affected by all of the kinds of influenza viruses circulating in a particular season. I would also note, and you may also want to comment again on the fact that with the influenza B viruses right now, there's a lot of interest in the potential role for a 4-valent vaccine. The vaccines we currently have available are trivalent or 3 strain vaccines covering one H3N2, one H1N1, and one B virus. But over the past 10 years or so, we have included the right B virus from the right family of the 2 families that are circulating right now about half the time. And so there is interest, and stay tuned, because I think beginning next season we will have at least 1 quadrivalent vaccine available.
We have a question from the room.
Yes, I was wondering how you balance 2 competing interests. You mentioned the overuse of antibiotics for the treatment of the flu. This doesn't help and creates other problems. But on the other hand, one of the leading causes of death is from bacteria pneumonia. So antibiotics would be used for the treatment of the bacteria. So how do you, on the one hand, not overprescribe for the antibiotics, but on the other hand, prescribe the antibiotics to treat the pneumonia so that people don't die? So if you have some recommendations on how to balance that.
So the influenza-like illness, or classic influenza, does not include pneumonia. It's an upper respiratory. Sometimes it can be a bronchitis kind of illness, but when someone comes in with classic influenza, they would not be coming in with pneumonia, and they wouldn't have an infiltrate on their chest X-ray and so on. So when it's influenza illness, we have laboratory tests that can help us confirm that. And if someone is coming in with symptoms, particularly severe symptoms, or if they're in a vulnerable population, we may prescribe antivirals for them. If they come in with a pneumonia, it's a very different scenario. Chances are their influenza viral shedding episode has already resolved by the time they've developed the bacterial pneumonia, and then we would try to diagnose that and treat it with antibiotics. So they're really 2 different time courses.
And just to briefly add to that, there are certain populations that are more vulnerable, that have underlying illnesses that make them more susceptible to develop pneumonias and secondary infections. And it's important to know about those people, immunocompromised, or people with other respiratory or cardiac illnesses, for example, or immunosuppression.
Another question from the room?
Yeah, so I was very impressed to hear about all of the misery, that flu brings us, and the millions of deaths and through the pandemics. So it sounds like it's a great public health goal to to kind of get that out of the human context. But I was sort of went through a thought experiment going even further. I mean, imagining you being perfectly successful, getting flu out of the public health issue, actually getting the flu virus entirely out of the ecological context. And so I'm wondering if there's a niche of virologists who look at flu and viruses in that larger context. And the question really is, if we got rid of it, if flu disappeared, would that actually in some ways have some negative ecological consequence? Or is it just good to get rid of it?
I'm going to defer to my colleague on this.
Yeah. So first of all, I think from a practical standpoint, I think that it would be absolutely impossible to eradicate influenza viruses, given the enormous depth and breadth of their reservoir in actually, it's unclear how many host species there are in, because there's actually never been active prospective surveillance in sort of every possible species to imagine. Unexpectedly, in small bats in Guatemala just last year, they found a new kind of influenza virus that's probably been there for forever. And so we have no idea what the whole reservoir is. So I don't think that's going to be practical. And it's not going to, even with the universal vaccine, probably not practical to completely eliminate influenza in humans, given this diversity of animal viruses that has the ability and propensity to move around between species. Certainly to at least achieve a goal in which we have vaccines that provide broader coverage, perhaps not a universal vaccine, but a vaccine that might provide coverage for more than a year or two, a longer amount of time, by improving the way the vaccine is made and moving toward a more universal vaccine is a better goal. But so I just don't see any way in which it could be eradicated.
Another question from the room?
Yeah, I'm Felix, a postdoc from Johns Hopkins. I recall the keywords in the presentation were surveillance and countermeasures. I'm sure you guys are familiar with the H5N1 controversy a few months ago, in which one side claimed that publishing the research related to the H5N1 was an issue of national security or terrorism, and the other side was claiming the importance of publishing that work. Can you guys comment on that?
Well, I can start. So influenza pandemics and new influenza viruses moving from animals to humans is clearly a very important and real threat. Pandemics have occurred in the past. They are going to likely occur again. H5N1 has been causing zoonotic, that is, infections from animals, from birds, predominantly poultry like chickens, to humans, at least since 1997. There have been several hundred deaths. I don't know the exact number on the top of my head, but more than half of the people who have been documented with those infections have died. So it has a case fatality rate potentially upwards of 50 or 60%, meaning that it's a very, very worrisome virus. If such a virus were to adapt to humans to be able to transmit efficiently person to person, it could cause a pandemic of unprecedented devastation. So there's a lot of concern, and therefore there's a lot of scientific interest about whether it's possible for such a virus like this to adapt to humans and to understand how these processes occur. So there are different ways to do this. One way is to do things that our lab has done, for example, is to go backward in time and look about host switch events that have occurred in the past and figure out how nature figured that out. And another view is to think about predicting, based on what we know, what could we say about the possibility in the future. So scientists with very legitimate scientific and public health interests did experiments to help try to understand that question. Those experiments have been and remain controversial, and there's no simple answer about that, but the motivation to do the experiment was truly related to this goal of trying to understand what the risk is. These experiments were done in very carefully controlled high containment labs, but since those experiments were done and those results were recently published, there has been a voluntary moratorium around the world of most of the labs involved in this kind of work, and this moratorium is still in place to give time for this kind of discussion to occur. And that no resolution of this question has actually occurred, that this moratorium is still in place.
Other questions? Someone's coming to the microphone. Oh, we have an internet question?
Yeah, we have another internet question asking about any clinical differences between influenza and SARS and how we can differentiate between the two.
I'm going to let you talk about SARS.
Okay, well, okay, so the question of the clinical thing, so I've not treated any SARS patients, but anyway, SARS, so SARS is a coronavirus. It's also an RNA virus. It's different than influenza, and also was a zoonotic infection, but you had, in this case, a coronavirus that adapted to humans. There are other coronaviruses actually that cause cold viruses and things, but SARS was a very serious infection. One of the very important differences was that the shedding tended to be at the end of stage of the disease. So actually most of the transmission person to person tended to be in hospitals where very sick people had been hospitalized and healthcare workers, rather than this kind of asymptomatic shedding before the onset of symptoms. So its control was actually easier because once you had a patient and could isolate it, classic quarantine could help control that, which is why in influenza I think it's very difficult to institute effective quarantine because of the shedding before symptoms. So I think that's one of the major differences.
And I think the SARS outbreak and also seasonal influenza and pandemic influenza also highlight the importance of surveillance to know what agent or agents you're dealing with generally in an outbreak setting. Sometimes symptoms can be very difficult to differentiate, particularly if people develop a febrile respiratory illness that may develop into a more serious syndrome. And so sometimes there isn't any perfectly predictive clinical syndrome without laboratory confirmation. And you know, what's circulating in the community or the context of an outbreak is very important in interpreting symptoms when people come in.
Another question from the room.
Great, thank you. I was curious if you could talk about surveillance a little bit more. I was at a public health meeting not too long ago. I'm a resident of Washington State, where the whooping cough epidemic is going on because of decreasing vaccination rates. And one of the things the Department of Public Health woman was saying was that their budgets have decreased to like 15% what they normally were because of federal budget cuts. So I'm curious if you could talk about surveillance, because it seems like we're kind of in this rising, you know, perfect storm for, you know, a great epidemic with, you know, antibiotic resistance and no surveillance in that. So I just wonder if you had any comments on that.
Well, I can start. So first, I want to say that human surveillance for influenza really is a great model of public health and something sponsored not only nationally but internationally through the WHO, that most countries in the world have national influenza centers that do surveillance in their country and pool those data to international centers and reference centers, and these data are used to make decisions about the vaccines both in the northern and southern hemispheres. So I think that certainly more surveillance should be done, but compared to other diseases, influenza has a really fantastic influenza surveillance system. I want to compliment that. In terms of surveillance in animals, there is surveillance, there's less surveillance than there is in humans, but there is surveillance in both domestic animals like horses and pigs and poultry and so on. There is some surveillance in wild animals. Of course, this is very limited and often done really on kind of shoestring kinds of budgets for people who are very interested in this question. There are more funds available for this now, realizing the importance of that, but there is no kind of systemic surveillance like we have in humans. And clearly, I think that more surveillance at the human-animal interface would be very important, not just for influenza, but for all the diseases like SARS or other things we've talked about are so often zoonotic diseases that come from animals into humans. So I think improving surveillance, especially in agricultural workers, would be an important goal.
And I think it's so interesting to think about heightened collaboration between the animal world and the human world in terms of surveillance and veterinarians and physicians and so on. I just wanted to comment also on the different types of surveillance that are available. And with influenza and with some other syndromes, we not only have active surveillance sites that submit specimens and so on, but there are some sites that just report influenza-like illness without laboratory confirmation. We plot hospitalization rates, we plot mortality rates, and so there are many different ways to engage in surveillance. Some of them are much more resource intensive than others. We have database systems now that are where we're developing algorithms to look for early syndromic events and syndromic surveillance. So I think surveillance is also evolving from just a laboratory-based confirmation of a particular agent to something that also looks at symptoms or hospitalizations and death and as a package, it's tremendously powerful, certainly for influenza.
Let's take one last question before we have to wind down.
I was hoping you could provide some metrics that quantify how effective the vaccines are. Two examples could be reductions in infection rates or reductions in shedding, something along those lines, or other areas.
Well, I'll be happy to jump in, and my colleague will also comment, I'm sure. So the influenza vaccines are what I think of as very good vaccines, but not perfect. And partly it's because they're trying to prevent infections to 3 different kinds of agents rather than just one. But if we look at different populations, there are different vaccines. In children, vaccination prevents infection and illness about 85% of the time, perhaps slightly higher rates of effectiveness or efficacy with a live attenuated vaccine, especially for young children. In younger adult populations, vaccine efficacy is probably around 55 to 65%, but there's a range from year to year. And in the elderly, we don't have many clinical trials that give the best kind of estimate for reduction in infections, but it's probably similar to, perhaps a little less than what we see in younger adult populations.
No, I agree with that. I don't have anything further to add.
I will perhaps also just, do I have time for one other comment? We used to talk about influenza vaccines in younger adult populations as being 70 to 90% efficacious, and we have more recently revised down those estimates in part because of better understandings of how certain laboratory tests perform and some challenges that some of the earlier kinds of laboratory tests present now with regard to how we interpret them. So we've revised some of those estimates down, probably 55, 65%, maybe 70% on average.
Thank you, Dr. Kristen Nickel, Dr. Jeffery Taubenberger, and all of you for your interest in microbes after hours. On behalf of the American Society for Microbiology, I'm Barbara Hine, reminding you certainly after tonight's program to get your flu vaccination and encourage your family and friends to do likewise. Good night.